I'm James Carey, Emeritus Professor of Entomology at the University of California, Davis. I originally presented this seminar on July 16, 2025, to the CSIRO group in Brisbane, Australia, led by Dr. Rick Van Klinken, Phytosanitary Risk Science Research Leader. Due to technical constraints at the venue, I was unable to record the seminar myself without considerable effort to reconfigure the setup for my own laptop. So instead, I've re-recorded the full seminar here in my home office in Davis. I've also added some additional material, including commentary on a 2014 meeting of the International Fruit Fly Group held in Bangkok, Thailand, where claims were made about our research that over 10 years later I still find disturbing. I also added several video appendices, including examples of fruit fly microdistributions, that is, cases where fruit flies were detected years, even decades later, in nearly exact same locations as earlier detections. Second, a detailed history of the medfly outbreak patterns in Southern California, material I didn't have time to share during the Brisbane seminar. Third, informed by analysis of all the historical data, my opinion of the likelihood of establishment for each of the 18 species ever detected in California. And last, a short list of key references for papers my colleagues and I have published that are related to the tephritid invasion of California. The reason I've taken the time to re-record and post this video is that I believe that California's long history with fruit fly outbreaks and the valuable insights gained from studying these patterns offer important lessons for entomologists facing similar challenges with invasive tephridids around the world. Specifically, there are four key reasons why these data are so significant. First, they're unprecedented in scope. Unlike most agencies, which tend to be secretive about both detections and specific locations, California's data is exceptionally detailed and transparent. Second, the diversity of species. The data include findings on 18 different tephridids, offering a comprehensive view of the problem. Third, extensive data with over 11,000 distinct detection lat-long locations. The breadth of this data set is really unparalleled. And lastly, historical depth. The data stretch back over 70 years to 1954, beginning with the detection of Mexican fruit flies, providing crucial historical context. With that background, I'll dive into the seminar. California's fruit fly crisis: global lessons in scientific denial and bureaucratic entrapment. Let me begin with a recent press release issued by the California Department of Food and Agriculture, CDFA, and the USDA on August 27, 2024. The announcement declared the USDA and CDFA are proud to announce that after a year of hard work, we have eradicated all populations of invasive fruit fly species in California. I've enlarged this statement here at the bottom of the slide so you can read it clearly. However, the very next day, on August 28, 2024, 61 adult medflies were confirmed in Fremont, a city in Alameda County, just south of San Francisco in Northern California. That was just the beginning. In September, additional medflies were detected. In October, it was an oriental fruit fly, not just once, but 3 or 4 separate times. In November and December, both medfly and oriental fruit fly were again confirmed. Then in February of this year, more medflies were found, and in early July, the oriental fruit fly made yet another appearance for the 57th year in a row. Then in late July, the guava fruit fly, Bactrocera correctus, was detected in San Jose. This reminds me of the saying, the operation was a success, but the patient died. In this case, the declaration of eradication was celebrated, but in reality, CDFA failed to eliminate the fruit flies. It met international benchmarks on paper, yet it failed to achieve the intended biological outcome. This is why I call it a crisis. California's fruit fly invasion, a 70-year struggle, is now approaching critical mass. It's not just a bureaucratic or scientific issue. It's economically devastating. California is home to the largest agriculture industry in the U.S., valued at over $55 billion annually. It's the 5th largest agricultural supplier in the world. The state grows more than 200 different crops, a large fraction of which are highly susceptible to multiple species of invasive tephridids. These are not just sporadic introductions. I strongly believe that some of these species, especially the oriental fruit fly, are now permanently established in the state. Their adaptation to California's environment appears to be complete. Historical background. I arrived at UC Davis in March of 1980 as a newly appointed assistant professor. Just a few months later, in June of that year, medflies were discovered simultaneously in the Bay Area and the LA Basin. The San Francisco Chronicle reported that the Bay Area infestation covered 1,400 square miles, spanned 8 counties and 43 cities, and affected 2 million people. Signs went up warning of quarantine zones. In backyards, residents and officials were pulling down fruit and bagging it, while insect traps were deployed. Aerial spraying began, prompting strong public protest as seen in these photos. In 1982, eradication was declared. After spending $100 million in the 1980 dollars, it's equivalent to about $350 million today. At the time, I was teaching insect ecology and incorporated the medfly into a demography handout. I developed a handout based on its life table, which eventually evolved into a Leslie matrix projection and a published article in ecological modeling. One of the key insights from that work, which I shared with the state's Medfly Advisory Committee, was that adults made up only about 5% of the total projected population. It's really a narrow band at the top of the population projection there you can see in the upper right. This meant that control efforts targeting only adults were limited by access to only 5% of the population. That paper led to funding from CDFA for demographic research on medflies in Greece, where I conducted host-specific studies and later published this in Ecological Entomology. From 1987 to 1994, during another major medfly outbreak, I was asked to serve on the California Scientific Advisory Panel for the medfly. My credibility stemmed from both my research in Greece and my position at UC Davis at the time. In the photo shown here, I'm standing with USDA scientists Carol Calkins, Roy Cunningham in the center, and Daryl Chambers, along with Dick Rice from the UC-affiliated Ag Experiment Station in the Central Valley. During this period, local newspapers were filled with headlines: "New fear on the medfly outbreak," "Medfly defies logic," "Medfly spreads to Orange County," and so forth. This was just a fraction of the hundreds of articles published statewide. On March 6, 1990, I was asked to testify before the Committee of the Whole of the California Legislature, that is to say, the entire legislature. The LA Times ran a front-page article quoting me as stating that the medfly was established. I may not have used the term endemic, which implies the species' native origin, but I argued it had become a permanent presence. This testimony was met with open hostility. This was no surprise to me, believe me. One editorial remarked that judging by the defensive reaction, one would think Carey had declared the world was flat and motherhood a subversive idea. Shortly after that, Science magazine covered the controversy in an article titled "Entomologist in the Medfly Maelstrom." USDA entomologist Roy Cunningham, chair of the advisory panel, dismissed my conclusions. Nevertheless, Science, the editor, contacted me and invited me to submit a research article, which was then published in 1991, titled "Establishment of the Medfly in California." It drew 13 letters to the editor, none supportive of my position, as might be imagined. Science published 2 of these letters along with my response. One letter from CDFA Director Henry Voss claimed incorrectly that a blue ribbon panel had concluded the medfly was not established. He criticized my use of incomplete data, which was not true, and asserted confidence that eradication had succeeded. History has shown otherwise on that statement as well. I sought USDA funding to continue my analysis, but reviewers claimed it would duplicate existing efforts and gave it low priority. I've never seen any effort on the part of the USDA to analyze these data. I also attempted to obtain internal funding from UC, but they told me essentially that I had to first bring CDFA and USDA around to my conclusions, that several tephridids were already established. Of course, neither agency wanted to have anything to do with acknowledging that fruit flies were established. These are some of the papers I've authored or co-authored on the fruit fly invasion, including the science article, 2 in ecology, 1 in ecological applications, 1 in biological conservation, and a proceedings paper from the Royal Society. One of the most disturbing episodes in my 45-year career was the agreement by 31 entomologists to sign on to the keynote address posed here at the International Fruit Fly Meetings in Thailand in 2014. Can Polyphagous tephritid pest populations remain undetectable over years under favorable climatic and host conditions? This was basically a setup by several of the leaders of the symposium, not to answer this question. They already knew the answer they wanted, but rather to give the impression that this was settled science, that there is no way that highly fecund fruit fly species could remain hidden for years and decades. This document was framed as a consensus statement presented as the authoritative view of the global fruit fly research community regarding the detectability and persistence of these pests. Here is a list of the signees from the 16 countries. Not a single one of the 31 entomologists who signed this statement had examined any of California's fruit fly detection data. There had been no modeling of the data set, no statistical analysis, no scrutiny of the trap records that extended over decades. Simply put, there was no data engagement at all. The statement by the symposium signees, this was in the document, any scientist who concludes that tephritid eradication efforts may not always be successful is not only unscientific, but irresponsible. Signing on to this keynote address document amounted really to signing a loyalty statement. The problem here is that the impact of consensus on the fruit fly persistence institutionalizes a contested assumption. It suppresses inquiry into cryptic survival. It reinforces a faulty eradication protocol. It closes the door on falsifiability, and it risks international groupthink. When declarations like this are accepted uncritically, they have serious consequences. An overview from my appointment till present. I was appointed in March of 1980. Here's a list of the protocols that have not changed over all those years. It's 45 to 50 years. The trapping protocols are the same. The trigger of when to launch an eradication program. The area around fines. The treatment. The one thing that has changed, there's no more aerial spraying. But nonetheless, the basic ground spraying protocols are pretty much the same. And how to end a program, when to terminate it. Basically, it's 3 generations by degree days without a detection. Despite the detection of 18 different tephritid species scattered throughout the state, the USDA, CDFA, and even the International Phytosanitary Commission have failed to revisit or revise the basic eradication approach. Programs end not because populations are demonstrably eliminated, but because the clock runs out. To close out this part, I've decided that for the past 50 years there are what I call the 3 laws of fruit fly intervention. First law is eradication programs are always 100% successful. Second law is new outbreaks are always due to reintroductions. Third law is silence the data, crush the dissent. These laws must change if California is ever to get on top of this slow motion developing catastrophe. Part 3, Tephritid Invasion of California. This is the story of California's long-running battle with invasive fruit flies. Between 1950 and 2020, California detected an unbelievable 18 different species of tephritid fruit flies. These include the Mexican fruit fly, the melon fly, the oriental fruit fly, the guava fruit fly, West Indian fruit fly, med fly, Caribbean fruit fly, peach fruit fly, Queensland fruit fly, striped fruit fly, pumpkin fruit fly, sapote fruit fly, Malaysian fruit fly, Bactrocera fascialis, which lacks a common name, the olive fly, the white striped fruit fly, and the tau fruit fly. This likely represents a global record for the number of invasive tephridids detected in a single region, even surpassing the number of pest tephridids present in virtually all tropical zones. Among these, 3 species stand out for their wide distribution. The Mexican fruit fly, detected in 77 cities, the Oriental fruit fly, 244 cities, and the medfly in 169 species. These numbers are current as of 2019, with probably more likely added in the past few years. California's fruit fly monitoring program began in 1910. Today it operates around 100,000 traps statewide, though not year-round, in all places. These efforts cost an estimated $25 million annually, totaling, in my estimation, roughly $1 billion over the past century. The database I've compiled contains 11,000 fruit fly records, each with date, location, that is latitude, longitude, species, and sex. I began compiling and analyzing these data in 1987 when I was a member of the CDFA MedFly Scientific Advisory Panel, and I've continued ever since. Here's an example. In September 1976, a single Oriental fruit fly was captured in Culver City. That record, like all others, includes precise coordinates and date. To keep the math simple, let's just round down to 10,000 individual fly captures. That's an average cost of $100,000 per fly. Of course, the system wasn't built for scientific inquiry, but for detection and protection. Still, these data hold immense scientific value. They're rich with insight into invasive dynamics and the efficacy of intervention protocols. The USDA has published maps showing where U.S. climates could support fruit fly populations, and most of the southern U.S. falls within these risk zones. The combined population of states within these fruit fly friendly climate states, including California, Texas, and Florida, and many others, is approximately 120 million. These are round numbers, with only 1/3, about 40 million, residing in California. If fruit fly propagule pressure were roughly proportional to population size, we would expect twice as many outbreaks outside California as within it. Even if we assume California is twice as vulnerable due to ecological, agricultural, trade factors, there should still be 1-to-1 ratio of outbreaks between California and the rest of the southern region. Yet across North America, while states like Florida and Texas have occasional detections, only California has documented 18 invasive species, with repeated outbreaks occurring year after year. Indeed, California accounts for 95 to 98% of all tephritid outbreaks in the U.S. In California, Oriental fruit flies have been detected every year for the past 57 years. This includes detections across the LA Basin, San Diego area, Central Valley, Bay Area, and coastal zones. A map produced by Ziwa Zhao illustrates this spread from 1960 through 2018. The LA Basin is particularly notable because it stretches 150 miles from Burbank to San Diego County and has had consistent and widespread detections. Between 2019 and 2023, detections surged, especially around Redlands, with over 650 Oriental fruit flies captured in that area alone. The Central Valley has seen detections of 6 different species: the Mex fly, the peach fruit fly, the med fly, melon fly, the guava fruit fly, and the Oriental fruit fly. The Sacramento and San Joaquin valleys are both affected, each missing one of the 6 species. Interestingly, despite the large number of migrant workers in the San Joaquin Valley, the Mexican fruit fly has never been detected there. This points to the possibility of persistence rather than multiple introductions. A key conceptual model I developed with Ziwa Zhao illustrates the growth eradication cycle. We call it phase 1. An incipient population is introduced and begins slow growth. Phase 2, it crosses a detection threshold, triggering eradication. Phase 3, post-eradication, a residual population remains, now more dispersed and harder to detect. This cycle repeats with shorter intervals between detections, suggesting incomplete eradication. Evidence-based policy implications. Policy-wise, CDFA still assumes all detections are new introductions, presumably patterned after the dynamics shown here: fast growth to detection, eradicate, repeat. But the evidence strongly supports a naturalization model like the one shown here. These populations have likely been present for decades, surviving at low, undetectable levels, until growing enough to trigger detection. Here are some questions my colleagues and I have attempted to answer. What is the probability of detecting a fly in a 10 by 10 grid throughout California? Statistically, the probability of detecting a fruit fly in any random 10 by 10 kilometer grid in California is about 4%. Once detected, the chance of a second detection in the same area jumps to 50%. And for a third detection, over 80%. In cities like LA, with over 23 known outbreaks, the persistence is virtually undeniable. Maps of outbreak risk show hotspots in the LA Basin, Anaheim, and Redlands area, with frequent and repeated detections. So what does a single fly detection mean? For the oriental fruit fly, it almost always signals a broader outbreak. Yet under older protocols, single medfly captures were deemed non-actionable, as seen in 1982, 84, and 86, leading to a massive outbreaks between 87 and 94. Absence of detections doesn't always mean absence of flies, of course. If a region has never had a detection, zero may mean zero. But in previously infested areas, a lack of detections may reflect populations below the detection threshold, not true absence. If California has successfully eradicated a single species, it should have been the oriental fruit fly. I use cause-specific elimination modeling. This comes out of health demography. Shows that eliminating the oriental fruit fly would have reduced the number of infested cities by over 100. From 366 to 256. In contrast, eliminating the mexfly or the medfly would yield smaller reductions. But of course, we wanted to get rid of all of them, but this is an exercise in if you wanted to do hair on fire level eradication, pull out every stop possible, and go after one of these flies, it would have been the oriental fruit fly. This book by Columbia professor Nancy Stepan is, in my view, the definitive book on eradication. Though the book is on epidemiology and focused on malaria, it highlights key principles on the eradication concept. As she notes, eradication means zero, 100% effective intervention in every affected grid. Anything less than eradication is control. So why and how difficult is eradication? Consider this hypothetical 100-cell infested region. If we have 95% effective treatment per cell, there's still a 99% chance the region remains infested. At 99% effectiveness for each cell, the probability is still nearly 2/3 that the pest remain present in the region. Even at 99.9% effectiveness, there's still a 10% chance of the region writ large is still infested. Now scale that to 1,000 or 10,000 cells or 100,000 cells, and you can see why true eradication really becomes hugely challenging. Importance of science and data. I included the concept of bureaucratic entrapment in the title because after encountering the term a few months ago, I realized it captures one of the central problems in fruit fly eradication. It refers to the rigid enforcement of detection-based protocols designed to preserve official declarations of success, even when the biological reality tells a different story. In the case of the oriental fruit fly, we've seen repeat detections year after year, yet the bureaucracy continues its wash, rinse, repeat cycle. While the protocol has likely slowed the invasion, it has also enabled the fly to spread throughout the state, reaching Redding, the northernmost city in the Central Valley. This is what I refer to as a failure of success, the ability to meet regulatory criteria for declaring eradication while failing to actually eradicate. Understanding the true nature of fruit fly outbreaks matters, not simply because we continue repeating the same brush fire response, but because once we accept that some of these species are in fact established, it becomes clear that closing introductory pathways alone is not a solution. Of course we must protect the pathways, but that won't solve the current problem if persistent populations are already present. Recognizing establishment changes everything. It allows us to begin predicting where and when outbreaks are likely to occur because risk is not evenly distributed across the state. We can develop contingency plans in anticipation of widespread detections, not just the rare massive outbreak like the one in Redding in 24, where over 500 oriental fruit flies were captured, but dozens of such outbreaks occurring across multiple regions in any given year. This is especially critical for high-value agricultural zones like the Central Valley. Acknowledging the reality of fruit fly persistence also means we can begin to provide meaningful warning to growers. These outbreaks are not bolts out of the blue. They are increasingly predictable. That opens the door to more strategic planning, including the development of fly-free zones and well-defined containment strategies as backup. Without recognizing these patterns, we're leaving both growers and the public unprepared. This is why analysis of the detection data is so important. From a scientific and strategic standpoint, we can deepen our understanding of the cryptic establishment, populations that persist at low levels and are difficult to detect, yet may be expanding slowly over time. Studying this phenomenon allows us to identify high-risk pathways, evaluate the actual effectiveness of eradication efforts, and improve models of range expansion and outbreak recurrence based on the historical record. Operationally, the data can help us improve protocol decisions. We can reassess whether current trap density and placement are sufficient, such as a long-standing standard of 5 traps per square mile. We can revisit assumptions about what zero detections really mean, especially in areas with past infestations. We can examine whether the current thresholds for triggering eradication are appropriate. We can explore what constitutes meaningful post-eradication surveillance, and we can revisit the central question, are we dealing with reintroductions or with populations that never fully disappeared? Even if we don't yet have all the answers, the data can move us closer to the truth. They offer far more insight than simply continuing to ignore the data. In closing, I want to emphasize that virtually all fruit fly entomologists who I know, who are involved in eradication programs, are deeply committed to the cause of protecting agriculture. I respect their intentions. I certainly do not take their criticisms of me personally, and I hope none take mine that way either. However, good intentions are not enough when data are ignored and scientific reality is denied. The long-standing pattern of dismissing persistent detections as reintroductions is really unsustainable. If California continues down this path, we risk facing a true crisis, one that could have been anticipated, understood, and mitigated. I'd like to thank Rick Van Klinken for hosting me in Brisbane, and 2 colleagues and co-authors on a number of key papers, Nikos Papadopoulos of the University of Thessaloniki and Zihua Zhao of China Agricultural University in Beijing. Thank you for listening. What follows are 4 appendices I mentioned at the beginning. Appendix 1 covers what I call micro-distributions. These are not detections at the city level, but rather right down to the block level. I examined initial discoveries and then later detections, often many years apart, that occurred in the same or nearby blocks. In virtually every case, with perhaps 1 or 2 exceptions, it is inconceivable to me that these later detections are due to chance reintroductions. You need to bear in mind that in a 100-cell grid, the odds that a new introduction would land in the same grid as a prior detection are only 1 in 100. Here's one example, the oriental fruit fly in Upland, California. Detections were first made in 1987. Notably, they were near the Ontario International Airport, something that likely has significance. Then, 21 years later, flies were again detected in exactly the same location. While it could be argued that this was a reintroduction, the absence of new detections to the north, east, or south of that area suggests otherwise. These flies may have persisted at subdetectable level for over 2 decades. Another example is in San Jose. During the Northern California outbreak of 1980 to 81, flies were detected in San Jose. A close-up of that area reveals a nearly identical pattern of outbreaks in 1992, 93, 2003, and 2007. Not only do these follow the same spatial footprint, but they also show expansion into adjacent zones. In Mountain View, also during the 1980 outbreak, detections were made. 8 years later, in 1988, another outbreak occurred very close by. Each detection point likely represents more than one fly. These later detections include both repeat and adjacent areas. Then we have Anaheim, significant because it was the first city in which oriental fruit fly was found, back in 1960. 45 years later, there was another detection just 10 blocks away. Whether the population sat undetected for that long is a question, but I believe it likely did. In one series of outbreaks, detections in 1976 and again in 2015 occurred just 300 meters apart, 39 years later. Appendix 2 covers the medfly history in Southern California. You've seen some of this already, but here I place it into a different context, emphasizing spatial and temporal patterns across a 36-year period. A reminder, the LA Basin is a very large region. I divide this history into several phases. Phase 1, the gathering storm, spans 1975 to 1988. In 1975, the first medfly detection in California occurred in Santa Monica and Culver City. It was followed by eradication. In 1981, new detections appeared in the San Fernando Valley, again in previously uninfested areas, with eradication declared. Single medfly detections in 82, 84, and 86, some in previous outbreak areas, were deemed non-actionable at the time. Then in 1987 to 88, new outbreaks appeared, some in older outbreak zones, and eradication was again declared. I refer to the 1989 to 94 period as Phase 2, out of control. I served on the scientific advisory panel for the medfly during this time. New and recurring outbreaks appeared in both new and old regions, 1991, 92, 93, and 94, with eradications declared in each instance. Remember, according to official protocols, eradication is declared if no flies are found for 3 generations based on degree day models, typically about a year. Next came what I call the area-wide management phase, starting in 1996. The sterile insect technique, SIT, was applied across broad areas. Outbreaks still occurred through 2004, but less frequently. These were largely in areas with prior histories of outbreaks. In 2005 through 2009, additional outbreaks continued, including in places like El Cajon in San Diego County, about 50 miles south of the main LA cluster. SIT was still in use, but outbreaks were still happening, though at a much lower rate. This timeline from Santa Monica in 1975 through 2009 shows a pattern, 60 different eradication declarations during that period, each considered 100% successful, yet outbreaks continued. To update this record, I examined medfly detections by county over the past decade. There were outbreaks in 9 of the last 12 years, spanning 8 counties. In 2014, detections were made in Los Angeles and Riverside. In 2015 and 16, they occurred in San Diego. In 2016, Solano County in Northern California joined the list, followed by LA and San Mateo in 2017. There were no detections in 2018, but they resumed in San Bernardino County in 2019. No medflies were detected in 2022, but in 2023 and again in 2024, there were finds in Los Angeles as well as Alameda and Santa Clara counties. Then in 2025, this year, there were detections again in Alameda and Santa Clara County. Appendix 3, Tephritid establishment likelihood. In light of the fact that most of these fruit fly species were virtually never detected elsewhere in the U.S., any repeat detection in California, whether after a year, a decade, or even longer, raises strong suspicions that these flies may have never been fully eradicated. My view is that the recurrence of these detections point to persistent low-level populations. Given that these same species do not repeatedly show up in Florida, Texas, and other fruit fly-friendly U.S. regions, nor in comparable surveillance efforts globally, California's recurring detections are unlikely to represent new invasions. So I've separated these out into 4 different categories. The species 100% established. First, the olive fruit fly. It's universally recognized as established, and it's managed as an IPM pest now. And the second is oriental fruit fly. It's not officially declared established, but recurring widespread detections makes its presence virtually certain. This has been over 57 years of continuous detection. Next category is species highly likely to be established. I consider the medfly. This is long-term repeated detections across the state. It strongly suggests that we have a persistent cryptic population. Next would be peach fruit fly. It reappears in both northern and southern California. Ongoing detections make eradication claims doubtful. The third is the guava fruit fly. It's recently found again in the Bay Area and Southern California. This is concerning due to its reemergence and spread. The next category is species to watch closely. I consider the Mexican fruit fly and the melon fly in this category. I thought at one point they were both established, but they have not been captured for a number of years, but still they are of concern. The next would be the Tao fruit fly. There is a major 2023 outbreak of this species. Just by sheer numbers, it is worth watching. And then in the same category would be the Queensland fruit fly again. In 2023, there was a major outbreak, and this one should be watched closely. The last category is species likely eradicated. All of those listed here have gone undetected for many decades, suggesting that successful eradication or environmental incompatibility took care of them. Here is Appendix 4. This is a list of key references my colleagues and I have published on the Tephritid invasion of California.